Welcome to G Fun Facts Online. Today we're diving into a story of incredible biological resilience. A story where an unassuming hero in the quest to conquer hearing loss resides not in a high-tech laboratory, but in the humble fish tank. The tiny, striped zebrafish, known to many as a common aquarium pet, holds the remarkable ability to do something humans and other mammals have lost: the power to regenerate the delicate sensory cells of the inner ear. The loss of these hair cells is a primary cause of permanent hearing loss, a condition that affects millions worldwide. By studying the zebrafish's innate gift, scientists are uncovering secrets that could one day lead to revolutionary treatments for deafness. This is a journey into the intricate dance of genes, cells, and signaling pathways that govern one of our most precious senses. Let's start with the scale of the problem. Hearing loss is a global health issue of staggering proportions. The World Health Organization estimates that by 2050, nearly 2.5 billion people will have some degree of hearing loss. For many, this is caused by the death of mechanosensory hair cells in the inner ear. These cells are named for the hair-like bundles of stereocilia on their surface. When sound waves enter our ear, they cause these bundles to bend, which triggers an electrical signal that our brain interprets as sound. The problem is, humans are born with a finite number of these cells. Once they're damaged or destroyed by aging, loud noise, certain medications, or genetics, they are gone forever. Our bodies can't make new ones. This inability to self-repair is a fundamental biological roadblock that makes most forms of this hearing loss incurable. Now, enter the zebrafish. This small tropical fish has become a powerhouse model for scientific research. Despite our obvious differences, humans and zebrafish share over 70% of their genes, making them surprisingly similar on a genomic level. This means many fundamental biological pathways are conserved, so studying them in fish has a high degree of relevance to us. What's more, zebrafish embryos are fertilized externally and are transparent, allowing scientists to watch the inner ear develop in real time within a living creature. A huge advantage over studying mammals where the inner ear is encased in dense bone. But the most compelling reason for the zebrafish's stardom in hearing research is its remarkable regenerative power. Unlike us, zebrafish can readily regenerate damaged hair cells throughout their lives, completely restoring their sense of hearing. This makes them the perfect model to unlock the secrets of auditory renewal. Like humans, zebrafish have an inner ear for both hearing and balance. This intricate system develops incredibly fast, with functional hair cells forming just 24 hours after fertilization. But they also have something we don't: a unique sensory system called the lateral line. This system is a series of sensory organs called neuromasts, distributed along the fish's body, which detect water movement and pressure changes. Each neuromast contains a cluster of hair cells that are functionally and morphologically very similar to those in our own ears. Because these neuromasts are on the outside of the fish, they are incredibly convenient for scientists to study hair cell damage and regeneration in a living animal, making the lateral line a primary workhorse in the field. So how does this miracle of regeneration actually happen? When zebrafish hair cells are damaged, whether by loud noise or ear-toxic drugs, a remarkable cascade of events begins. Within hours, the process of regeneration kicks off, and in just a few days, the area can be fully repopulated with new, functional hair cells, leading to a complete recovery of hearing. The key to this feat lies in a population of cells that, in mammals, have lost their potential: the supporting cells. In both zebrafish and mammals, hair cells are surrounded by these supporting cells, which maintain the structure of the sensory tissue. In mammals, when a hair cell dies, the supporting cells just stay there, forming a non-functional scar. But in the zebrafish, these same cells are the source of new life. Upon damage, the zebrafish's supporting cells spring into action using two main mechanisms. First is direct transdifferentiation, where a supporting cell directly transforms into a hair cell without dividing. It's a remarkable feat of cellular reprogramming. The second is mitotic regeneration, where a supporting cell divides, and one or both of the new daughter cells then differentiate into hair cells. This allows for a more substantial repopulation after extensive damage. Recent research shows the zebrafish uses a sophisticated dual approach. After damage, there's an initial burst of supporting cell division to expand the pool of potential replacements. Then, new hair cells begin to emerge through the direct transdifferentiation of these new progenitor cells. This elegant strategy ensures a steady supply of new cells to restore function. To truly understand this, we have to look at the molecular toolkit for regeneration, the symphony of genes and signaling pathways that orchestrate the entire process. A key player is the WNT signaling pathway. WNT is a well-known regulator of cell proliferation. In zebrafish regeneration, the WNT pathway is crucial for promoting the division of supporting cells, leading to a more robust response. Interestingly, it isn't activated immediately but rather at later stages, suggesting it's not the initial trigger but a key driver of the proliferative phase once it's started. Then there's the Notch signaling pathway, a master regulator of cell fate. During normal development, Notch acts as a form of lateral inhibition. A cell destined to become a hair cell signals its neighbors to become supporting cells instead, creating a perfect pattern. During regeneration in zebrafish, the Notch pathway plays a critical role. Immediately after damage, Notch signaling is temporarily turned down. This release of the Notch brake is a crucial step that allows supporting cells to re-enter the cell cycle and begin regenerating. Later, as new cells are forming, Notch is reactivated to ensure proper cell fate decisions and control the number of new hair cells being produced. Another important player is the fibroblast growth factor, or FGF, signaling pathway. Its role is complex and seems to depend on context. Like the WNT and Notch pathways, FGF signaling is also downregulated right after hair cell damage. This suggests that a coordinated, temporary shutdown of these key developmental pathways might be a prerequisite for initiating the regenerative program. At the heart of these pathways are transcription factors, proteins that turn genes on or off. Groundbreaking recent research identified two families of these, the Sox and Six families, as the master conductors of zebrafish hair cell regeneration. Using advanced single-cell genomic techniques, scientists have mapped the entire process. They found a two-step mechanism. First, following hair cell death, Sox transcription factors, particularly one called Sox2, are activated in supporting cells. This initiates the response, causing the cells to become more progenitor-like. Second, as these progenitor cells differentiate into new hair cells, the Sox and Six transcription factors work together to guide the transformation. The discovery of this Sox6 regulatory network is a major breakthrough, providing a specific set of molecular targets that could potentially be manipulated in the mammalian ear. Beyond the genetic code, there's another layer of regulation called epigenetics. These are chemical modifications to DNA that act as a form of cellular memory, helping to lock in a cell's identity. Research shows that in mammals, the genes required for hair cell development are epigenetically silenced in supporting cells after birth. This epigenetic block is a major reason our supporting cells can't regenerate hair cells. In contrast, the epigenetic landscape in zebrafish supporting cells is more permissive, allowing the hair cell genetic program to be reactivated after injury. This brings us to the central question: why can't we be more like a zebrafish? The reasons are complex. As we've seen, our supporting cells largely remain quiescent, partly due to the active suppression of cell division. The molecular signaling environment is also less helpful. That Notch brake seen in zebrafish doesn't release in the same way in mammals. And, of course, the epigenetic silencing of key hair cell genes poses a huge barrier. So how do we translate these lessons from the fish tank to the clinic? The ultimate goal is to develop effective therapies for human hearing loss. The zebrafish model is already proving invaluable. for example in drug discovery. The small size of zebrafish larvae makes them ideal for high-throughput drug screening. Scientists can test thousands of compounds to see if they can protect hair cells or promote regeneration. This has already led to promising otoprotective compounds. One such compound, ORC 13661, identified through zebrafish screening, has successfully completed Phase 1 clinical trials and is being developed to prevent hearing loss in patients receiving certain antibiotics. Gene therapy is another promising avenue. By understanding the roles of genes like those in the WNT, Notch, and Sox families, scientists can develop strategies to deliver them to the inner ear. One exciting approach being explored combines a drug-like cocktail to activate specific pathways with the delivery of a key hair cell transcription factor called ATO1. This combination was able to induce the regeneration of new hair cells in an adult mouse model, demonstrating the potential of a multi-pronged therapeutic approach. And the research continues to expand, now exploring the role of the immune system. After damage, immune cells called macrophages are recruited to the site to act as a cleanup crew. Their precise role in regeneration is still being investigated, but it highlights the complexity of the regenerative environment. Despite the incredible progress, major challenges remain. A key hurdle is translating these findings to the much more complex adult mammalian inner ear. Often, newly regenerated hair cells in mammal models are immature and not fully functional. Future research must focus not just on creating new hair cells, but ensuring they mature properly and integrate into the auditory circuitry. The humble zebrafish has provided us with a biological blueprint for hearing regeneration, a roadmap to a future where deafness may no longer be a permanent condition. By meticulously deciphering the lessons encoded in this tiny fish's genome, scientists are piecing together the intricate symphony of genes and cells that can orchestrate the renewal of our hearing. The journey from the fish tank to the clinic is long, but the promise of restoring the gift of sound to millions is a powerful motivator. It's a testament to the power of basic research to illuminate the path toward a future filled with sound.